Bio 100, Chapter 10, Cell Reproduction. Alrighty, so we're kind of going back to some of the basics now that we've talked about other things so that we can get into what cell division is and then talking a little bit about how that can lead into cancer and those kind of things. So starting with the basics, cell division has two different functions, right? As multicellular organisms, which is humans, we use cell division for growth, maintenance, and repair of cells and tissues. And single-celled organisms can use cell division to reproduce, which we'll kind of touch on just because I think that it's useful to know a little bit about single-celled organisms as humans, because this, for the most part, and how we will relate to it, is talking about prokaryotic cells, which is bacteria for the most part. Okay, so cell's DNA makes up its genome. So in prokaryotes, like bacteria, the genome consists of one double-stranded circular DNA molecule, and that's in something called the nucleoid. And then there's additional smaller loops of DNA called plasmids that are present. In the eukaryotic genome, which is humans, as well as other plants and animals, we actually have several double-stranded DNA molecules in the form of chromosomes. So the number of DNA molecules or chromosomes in the cell varies among the species. In humans, we have 46. So we have 23 that come from mom and 23 that come from dad. So you'll often hear it as 23 pairs of chromosomes or 46 total. Two of those chromosomes are the X and the Y chromosome, which are our sex chromosomes that determine what sex you are, whether it's male or female when you're born. So within a species, the chromosome number is consistent, but can vary with developmental stage or specific cell type. There's also things that happen like having an extra chromosome. Extra chromosome. So extra chromosome would be like folks that have Down syndrome. It's referred to as trisomy, and that is one example of it. Okay, so somatic cells and reproductive cells. So somatic means body, reproductive cells obviously are your reproductive organs in most cases. So somatic cells typically have two matched sets of chromosomes. So when we're talking about our 46 total chromosomes, 23 coming from mom, 23 coming from dad, it's really 22 somatic cells. And these are what we call diploid cells, 2N, which means that they have two matched sets of chromosomes. Whereas gametes, which are reproductive cells, are often called our sex cells, which would be eggs for females, sperm cells for males, have half the number of chromosomes, and so they are what are referred to as haploid, or 1N. So when we're talking about diploid, that would be somatic body cells. Haploid would be our gametes or sex cells. Arranging the chromosomes by size produces what's known as a karyotype. So chromosomes or homologs that pair in reproduction of diploid cells are described as homologous. Some genomes have pairs that don't match. So for example, X and Y chromosomes in humans, if you're a male, or XX in females, these are what we refer to as heterologous pairs. So homologous is basically the same, right? Diploid cells, when we are going through cell reproduction, we're going through a process called mitosis. And when we're making diploid cells, we're basically just making exact copies of the cell. So we're just going through cell reproduction and division and making exact copies, whereas when we are making haploid cells, or sex cells, those are not exact copies. Those are going through what's called meiosis, and meiosis has meiosis 1 and meiosis 2, which we'll get into in a little bit. So compared to prokaryotes like bacteria, eukaryotic DNA has to be condensed into compact chromosomes in order to fit into the nucleus. So you can see over here a DNA double helix. You've probably all heard of the DNA double helix. And basically what it is we have see this light blue here is the backbone that's made up of phosphates. And then the little rainbow colors in between, those are base pairs. So that's like adenine paired with thymine or cytosine paired with guanine. These are what make up your DNA. Okay? So in order to fit all of this into tiny little compact chromosomes that fit into the nucleoid, you'll see you have this double helix, and then that gets wrapped around something called a histone. And then these nucleosomes coil into a chromatin fiber, and there's further condensation of chromatin, and then we have the duplicated chromosome here. So just all of this little amount you can see fitting into each one of these stages. And the way that we package this accomplishes in several different ways. So short stretches of DNA, again, wrap around those things called histone proteins. It's like a string of beads wrapped around each other. And then the histone DNA complex, which is the bead, is called a nucleosome. And then the connecting DNA, or the string, is called linker DNA. The structure will then coil to form a chromatin fiber, and these fibrous proteins further pack each chromosome. So I will post this video you can look at for chromosome packaging, how that happens, and get a little bit of a deeper look into it. But let's go through and actually talk about what the cell cycle is. Cell cycle is an ordered series of events in the life of a cell. And if we are talking about somatic eukaryotic cells, so where we're going from diploid to diploid, we're just going to make exact copies. We're going to go through something called mitosis. Okay. Keep in mind when we talk about this, we generally refer to this whole thing as mitosis, but really mitosis is in here. So before mitosis can begin, we have to go through something called interphase. So if you're ever looking under a microscope and you're looking at whatever it might be, like onion tips or cell tissues from something, you'll see thousands of different cells, and some of them are in different states of division as well. Like say if you're looking at an onion root tip, I know that sounds weird, but that's one of the most basic things to look at to get a really good feel for mitosis. If you're looking at that, onion root tips can grow really quickly, and so you have mitosis going on really frequently. And when you're looking at these, you'll see a majority of the cells are actually in this thing called interphase, because interphase takes a lot more time, mitosis, and then subsequently cytokinesis, which we'll talk about these two things in just a second here. But these two things happen relatively quickly within this whole process. So when you're looking at cells, you'll see many more cells in this interphase phase just because it takes so much more time to go through. In interphase, there's 3 different stages. So we have G1, which is basically where we've accomplished, or not accomplished, but we've said, okay, hey, we need to divide, right? And there's different checkpoints within mitosis as well. So technically the first checkpoint is like right here, and it says, hey, we need to divide for whatever reason, right? So once your cell decides it needs to divide, everything's going to get bigger. It's just going to grow more cytoplasm and make room to essentially grow another cell or reproduce. Then it's going to go through the S phase of interphase. S phase is where the DNA is synthesized. So this is where we're making an exact copy of the DNA. And before we get into this, our last checkpoint here within G1, which is our first phase of interphase, is basically saying, are there any issues with the DNA we're going to copy? If it's a no, then we're going to go into the S phase. We're going to copy all of the DNA. And then once we get into the next phase, the last phase of interphase, G2, we have one other checkpoint that says, okay, is all of the DNA that we copied look good? Are there any mistakes where we would need to kill off this cell because it's going to be a problem later? And if we pass that checkpoint and it says, nope, good to go, then we're going to continue to grow just a little bit more, and then we're going to go into the mitosis and cytokinesis phases. Okay, so again, the cell cycle has the 2 major phases. First is interphase, which you'll see here in blue, and that's the time for normal growth and preparation for cell division. And then the mitotic phase, which is smaller, we often refer to it as M phase. That's in red. That is where the replicated DNA and the cytoplasm are split, and the cell actually divides to make 2 new daughter cells. It's called daughter cells, even though that's not It does not indicate whether it's a male or female, but they're called daughter cells and that they're just identical cells. Okay, so again, interphase is the longer part of it, and it's happening in 3 stages. So we have G1 first, and the cell is basically just active, right? It's getting bigger and it's preparing for S phase, which is when DNA synthesis occurs. So identical copies of the DNA molecules or sister chromatids are joined at a thing called the centromere, and then the centrosomes produce mitotic spindles to move the chromosomes and help organize cell division, essentially. And then in G2, which is the end of interphase, energy is replenished, organelles reproduce, and the cytoskeleton breaks down. Mitotic phase involves several steps. So there's something called karyokinesis, also called mitosis. That's nuclear division. That's the first step. Within mitosis, there are 4 different steps. So this can get a little confusing, but within mitosis, you have prophase, metaphase, anaphase, and something called telophase, which is often associated with the second portion called cytokinesis. And cytokinesis is the very end of it here, where we're actually just dividing these 2 cells into 2 different cells. The cytoplasmic components physically separate into our 2 daughter cells here. So here's the stages of mitosis. Again, we're going through interphase, we're ending with G2, then we're going into prophase, which is the first part of mitosis. You'll see these early mitotic spindles forming these centromeres that are essentially holding these chromatids together. Fragments of the nuclear envelope. So the thing that's holding your DNA, the nucleus, the nuclear envelope surrounding it is starting to kind of disintegrate, essentially. And then we go into something called prometaphase, or I usually just refer to, honestly, metaphase. Metaphase is pretty easy to pick out because metaphase just means that all of the chromosomes are lined up, or chromatids, I should say. Chromosome would be 2 chromatids together, and now when they're just single, that's a chromatid. So all of them are lined up here right in the middle. Then we go through something called anaphase, where these little mitotic spindles connect up to the centromere. So like the middle of these little chromatids, it's going to connect up and it's going to pull them apart to each pole, basically, because this is how we're then going to create our 2 new daughter cells. So then we go into telophase and cytokinesis. So telophase is the last part of mitosis. Cytokinesis is technically separate from mitosis, and that's when we're separating the 2 cells. So that's kind of like I always think of it as like the karate chop of this, where we're karate chopping right in between here, and that's connecting both of these cells to make 2 new identical cells. Okay, so going into it a little bit more detail, again, we're starting with prophase and mitosis. That's where our nuclear envelope is breaking down. Our membranous organelles, like our Golgi complex, our endoplasmic reticulum, are dispersing towards the edges of the cell. Our nucleolus is going to disappear, and the centrosomes are going to begin migrating to the poles. Those little microtubules that are helping pull those chromosomes or chromatids to either side are starting to form, and then the sister chromatids are coiled together a little bit more deeply. Prometaphase, so this is kind of the in-between prophase and metaphase. Again, I normally just kind of skip to metaphase, but the book kind of goes through it like this, so we'll stick with that. But prometaphase, sister chromatids develop a protein kinetochore, which is right here in the centromere region, which attaches the chromatids to the spindle microtubules. So you can see the centromere here. This is what these microtubules, these like little lines coming off, are attaching to pull these guys off to either end. Okay, so these are the sister chromatids here, and they're basically just going to be separated, and that's when they're a chromatid. Okay, so then it goes through prophase, metaphase, or prometaphase, metaphase, anaphase, and now we're actually getting into the actual cell division, which is cytokinesis, and where we're going to physically separate the cytoplasmic components into the 2 daughter cells. Metaphase, I think that my slides might actually be a little bit out of order here. But metaphase, again, the chromosomes are lining up along the metaphase plate, and sister chromatids remain attached by cohesion proteins. So you can see they're still attached here, but these little microtubules are getting ready to go in and grab these now that they're nice and lined up in the middle to pull them over to the side in anaphase. So the cohesin proteins degenerate, allowing the chromatids to separate. So these are our sister chromatids, right? Now they're separated into just chromatids, and they move in opposite directions towards the centrosomes to which their microtubules are attached. So you can see they're attached over here, and then the cell is elongating even further, and then we're going to come through the middle of it here to cut this. Telophase, this is where the chromosomes have now reached opposite poles and begin to decondense or unravel. Spindles depolarize into tubulin monomers that will form cytoskeleton components for the daughter cells, and we see the nuclear envelope starting to reappear. So this is basically forming our nucleus that's going to contain all the DNA. And we can see the nucleosomes appearing within that nuclear area as well, these little things right here. So now we've basically got all the parts to make our two cells. We just need to physically karate chop it, which is where the kinesis comes in. And that does differ between plants and animals. Not super important, just of note because it's in your book. But you'll see whenever you're looking in the microscope and you see kind of this like little furrow here, I usually consider this like telophase / cytokinesis because honestly, especially under a scope and if you're not watching this in real time, this can be kind of hard to differentiate because this is like the very last moment before we're karate chopping to form these two daughter cells. So this would be cytokinesis technically. In plants, we actually have something called a cell plate. So it's a little bit different. I wouldn't worry about the plants too much. We're just focused on the human cell division. Okay, so again, within this cell division, we have these things called checkpoints. And basically, it's like quality control, right? And why do we have quality control? Well, we have quality control because we want to make sure that our cells are good. If our cells aren't good, we're going to introduce them to something called apoptosis, which is programmed cell death or cell suicide. And the reason we would want to do that is if we say had a bad mutation, or if we didn't copy our cells correctly, or whatever it might be, or if they're cancerous cells, right? We don't want to keep reproducing these cells if they're bad, essentially. So any mistakes that affect function, like mutated chromosomes or the wrong number of chromosomes, really, are regulated at these three checkpoints. So we have our first checkpoint. Remember I said like our first checkpoint's technically over here. That's just basically the trigger to divide, but usually what we consider the first checkpoint is here near the end of G1 where we're saying, okay, does our DNA look good enough that we want to go through this process? And then at G2, to mitosis transition, we have our next one that's saying, okay, was the DNA replicated properly? And then in metaphase of mitosis, I think I forgot to mention this one earlier, but we have another checkpoint here that's basically like all of our chromatids are lined up and they're ready for anaphase. So we want that to basically regulate cancer, right? Or to regulate our cell division. And cancer really is just unregulated cell division. So a lot of times why cancerous cells are reproducing so quickly is because they're skipping these cell cycle checkpoints. They're skipping quality control. Okay. So how else do we control the cell cycle? We have these cell checkpoints that tell us yes or no, we shouldn't keep dividing. But we also have external triggers that can initiate or inhibit the cell cycle, like death of nearby cells, if there's release of something called growth hormones, which is literally exactly what it sounds like, telling you to grow, right? Or if there's cell crowding. There's also internal factors that can regulate the process too. So again, going through the checkpoints in a little more detail, G1 checkpoint is going to determine whether all conditions are favorable for cell division. Again, is there any sort of genomic DNA damage? If it does not meet all the requirements, it's not going to be allowed to enter the S phase. So if the conditions aren't met, it has two different options. It can either stop the cycle and try to fix the problem, or it'll enter G sub O, G initial, and wait for signs that conditions are better. So G sub O is right before G1, and we don't really talk about it too often because it's kind of like the waiting period, like the the place where you're just kind of in limbo. B2 is preventing entry into the mitotic phase if certain conditions aren't met. So cell size, protein reserves are checked again. Most importantly, though, we want to make sure that our chromosomes are replicated and that the replicated DNA is not damaged. If there are any problems detected, the cell cycle is going to stop, and it's either going to try to complete the DNA replication or repair the damaged DNA. And then the checkpoint, the last one that is at the metaphase, it's occurring near the end of metaphase, and it's determining whether all the sister chromatids are correctly attached to the spindle microtubules. These are the things that are going to pull them apart towards the poles for anaphase. So this is kind of known as the spindle checkpoint. Again, the cycle is not going to proceed until the kinetochores of each pair of sister chromatids are firmly anchored at least 2 spindle fibers, and failure to correct this could lead to something called non-disjunction of chromatids. Non-disjunction basically just means that there's an abnormal number of chromosomes in all the cells, and that can lead to different things like, say, Down syndrome. Two groups of intracellular molecules are regulating the cell cycle. We have positive regulators that are promoting the movement to the next step of the cell cycle. And then we have negative, right, which is the negative regulators are stopping the advancement of the cell cycle. We also have silins and cyclin-dependent kinases, CDKs. The level of these proteins fluctuate predictably over the cell cycle, so this is kind of going into more of the external and internal signals that can trigger increases in these cyclin protein levels that essentially help tell you which phase to go into at what times. So a little bit on how positive regulators work here. You can see like our cyclin will bind to the CDK and it's going to become phosphorylated. I'm not super concerned about you understanding really what that means, but it's going to phosphorylate the target protein, which is then going to activate the protein, and that's what's going to advance the cell cycle. So basically it's just saying like, hey, we're activating things slowly and we're wanting to progress into the cell cycle where we can divide. Negative regulators are a little bit different. The best understood are retinoblastoma protein, and you can see this in different pages in your books, but these act primarily at the G1 checkpoint. So when we're first checking, like, is everything good? Are we ready? And you can see the unphosphorylated RB binds to a transcription factor E2F, which cannot bind the DNA, and it's going to block something called transcription, which we'll get into more at a later time. So basically what to know about this is that if this happens, we cannot grow the cell, really. We can't synthesize DNA. So what will happen is cell growth is going to trigger the phosphorylation of RB. So phosphorylation just means we're going to add phosphate groups here, right? And then that's going to release the E2F, which is going to then bind to the DNA and turn on something called gene expression. And that's what's going to get the cell cycle to keep going because we turned it on down here, even though we had to disconnect this. So it's a little bit different, but again, it can stop the cycle at certain points, or have it continue on in a different way. Okay, so getting into cancer. Cancer covers lots of different diseases characterized by uncontrolled cell growth. Really, at the heart of it, what cancer is uncontrolled cell growth. And it begins with a gene mutation that results in a faulty protein that regulates cell reproduction. So we'll get into this a little bit more in genetics, but basically your DNA is the blueprint for what proteins your body needs, right? And if your DNA is damaged, it is either not going to make proteins that your body needs, or it's going to make the wrong proteins for your body. Either way. And so cancer is essentially doing parts of that. It's basically not controlling your cell growth, and it's going through skipping all those checkpoints of quality control and just copying all of those cells that were potentially already mutated. So that's also how you can get a tumor, right? These things happen really quickly. Tumors result when reproduction of mutated cells surpasses growth of normal cells. And again, the way that they're doing this is they're skipping all those checkpoints. So we have something called proto-oncogenes, or like oncologist, right? That's meaning cancer. Proto-oncogenes are normal genes that code for positive cell cycle regulators. So when these genes mutate in certain ways, they become oncogenes, which is cancerous, right? So you can see a normal P53. So DNA damage, cell cycle abnormalities with hypoxia. We can go into cell cycle arrest and go into DNA repair and then restart the cell cycle. Or if we stop the cell cycle, we could just do apoptosis, which is programmed cell death, right? And that's ideally what we want to see. We want to see, oh, okay, we see this weird abnormality, so we either want to go back through and make sure that we fix it before we replicate, or we want to just kill off the cell, right? So that's what P53 is going to do. If the damage can't be repaired, then it's going to go through the cell death. If it's mutated now, so now we have DNA damage, there's cell cycle abnormalities, but it's going to skip those checkpoints and the cell cycle is going to continue, and that's when the cells are going to become cancerous, right? It's just going to continue to divide, which will often result in cancer. Kind of a lot more than we really get into or really have time to get into in this class, but there are things called proto-oncogenes and tumor suppressor genes. Proto-oncogenes basically is like the go signal. So you can see the mutated one here, and it's going to continue. Proto-oncogene is the go signal. So the mutated one just says, it's all good, keep going, go, 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 and that's why you start building up all of these cancerous cells so quickly. And that's why if you ever hear anyone diagnosed with cancer, they say, oh, we really want to get it before it metastasizes and grows and continues to grow to different organs or different parts of your body. That's essentially what they're wanting to stop, is this unregulated cell growth. So the other one is tumor suppressor genes, and these are just segments of DNA that code for negative regulator proteins. So remember, we have the positive regulator and the negative regulator. So proto-oncogenes is the go. Tumor suppressor genes now would be at the stop, our negative regulator. And when these are activated, they prevent uncontrolled division. So cells that have a mutated form of a negative regulator might not be able to stop the cell cycle if there's a problem, which is why you get these tumor suppressor genes that would normally tell you to stop. They're a good thing, right? If that is mutated, again, then it's turned on and it's telling you go, 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 and that's when you can get things like tumors or cervical cancer. Those are mutations in tumor suppressor genes. Okay, I know that was kind of a really brief intro into cell division and cancer. Just to mention prokaryotic cell division, again, this is mostly bacteria. They do propagate by something called binary fission, which basically means that they're just producing new individuals asexually. So they don't need to go through the extensive cell cycle that we do in mitosis or meiosis, which is where you're reproducing sex cells. They're just reproducing by binary fission, which is really fast and can go through hundreds of thousands of bacteria, which is why bacteria can grow so quickly in so many different places. So I won't really get into this. I just think it's kind of interesting because it is different than mitosis. It can happen much quicker than mitosis. And it's something worth noting in human biology just because bacteria obviously does affect us. Bacteria is the cause of many different ailments in humans. And it's the reason why you would be prescribed antibiotics, let's say. So keep that in mind if you do have like a virus like flu or COVID or something like that. Your doctor should not be prescribing you antibiotics. Antibiotics are specifically targeted to kill off bacteria, and they're doing so by going after the cell wall that's in bacteria, and that does not exist in humans. Humans do not have cell walls in any of their cells, but bacteria do. So antibiotics are working on that. And again, you want to do that so that you can stop this binary fission and this just uncontrolled cell division in bacteria. That's all I'm really going to mention on binary fission or cell um, division in bacteria. I just think it's interesting and worth noting to be mindful of as a human trying to avoid bacterial infections.